The overall goal of this protocol is to generate protein based on the extracellular domain of myelin oligodendrocyte glycoprotein, or MOG, which can be used to induce experimental autoimmune encephalomyelitis, or EAE.
This protocol makes it possible for any lab to make MOG protein in order to use as an antigen to induce EAE. The main advantage of this protocol is that it doesn't rely on specialized protein handling equipment, but instead you can use standard equipment found in almost any immunology lab.
Well, this protocol was optimized for our MOG-tag protein. Based on the mouse version, it should work for other MOG protein expression systems that incorporate a histag for purification.
Inoculate 5 milliliters of sterile lysogeny broth with a BL21 MOG-tag glycerol stock and incubate it overnight at 37 degrees Celsius and 200 RPM. Add 500 microliters of 100 mg per milliliter ampicillin to each of 2 500 milliliter flasks containing 500 milliliters of sterile LB. Transfer 1 milliliter of the overnight culture to each of the 2 flasks of LB broth. Incubate at 37 degrees Celsius and 200 RPM for 5 hours, or up to an optical density of 0.6. Once the desired cell density is reached, add 0.5 milliliters of 1 molar IPTG to each culture flask. Incubate the flasks at 37 degrees Celsius and 200 RPM for 4 hours, and then at room temperature and 75 RPM overnight.